partner with me. We founded Cell Surgical Network, and originally the California Stem Cell Treatment Center. We've been doing safety and efficacy studies for the last 6+ years on autologous stromal vascular fraction. And we're starting to understand that the nature of certain conditions requires a little bit different approach to cell therapy. And we understand that stem cells are going to play a role in the next few decades in the evolution of personalized medicine. And I'd like to just speak a little bit today about what personalized medicine means and how we all might be part of personalized medicine. I think it's a little bit of a buzzword. You hear a lot of the term personalized medicine being thrown around, but it's often not defined, and it's really very interesting. I started reading about it and learning about it, and I'm going to share a few slides with you, and then I want to talk a little bit about some investigative work we're doing on how we might get ourselves involved in developing personalized medicine. I'm just having a technical problem. 1 second. This should be advancing my slide. Is there? Do
do it in slides.
Is it going to work now? Okay, thanks. Okay, so simple Wikipedia definition. It's personalized medicine is using medical decisions and practices and interventions and products tailored to an individual patient and based on their predicted response or on their predicted risk of a disease. Other terms might be precision medicine, P4, stratified medicine. They all really mean the same thing, and the definition that I found in my reading that I liked was just simply stratifying patients based on diagnostic and informatics, which is basically testing and data, to help select the optimal therapies, and based on the person's individual genome or molecular or cellular analysis. And again, the emphasis seems to be on, since it's on stratifying, the emphasis seems to be on really screening and detection, not so much on treatment. So today, currently in use, we see examples of personalized medicine. In the little diagram, this is just sort of a schematic showing how different patients might respond to different medications. We do need to understand better which patients will find that certain meds are more effective and that certain medications are safer. And we're starting to look at this in a very meaningful way. One of the other ways we're using personalized medicine is in selecting oncology agents, chemotherapy agents. We will often biopsy or use the biopsy of a tumor to evaluate the genomics of the tumor, look at the mutations, and look at the proteins expressed and see which drug they might do best with. Maybe another example of personalized medicine is the compound pharmacies, which are a little bit more tuned in. Instead of having a pill in a certain dose, they'll look at different ways of delivering the medication, different ingredients and routes. Just interesting, back to the point about medications, it turns out there's only 2 human genes that affect how we process most of the drugs on the market. So, but there are big individual differences. This has led to the field of pharmacogenomics, and the FDA has addressed pharmacogenomics in a meaningful way. In October 2013, they released a little treatise called Paving the Way for Personalized Medicine, and it was sort of a game plan in which they outlined steps that a regulatory agency would take to start integrating some of the genetic and biologic information in clinical drug development. And they started creating a genomic reference library as basically a large database to guide them. This is sort of a well-known article written in 2013 called The Faces of Personalized Medicine. And it defined pharmacogenomics well in that they used the phrase pharmacogenomics is the right therapy with the right drug at the right dose in the right patient. And I think that sort of describes very well the direction of pharmacogenomics and that example of personalized medicine. So the personalized medicine has tremendous potential. We talk a lot about it, and we have some solid real-life examples. But one of the issues, or maybe you could say one of the problems with personalized medicine, is that it still heavily emphasizes on the treatment side, algorithmic thinking and treatment algorithms. So in terms of detection and screening, it's maybe a little bit more developed. But on the other side, the treatment side, it really boils down to algorithms. So if you try to evaluate how to treat a patient, you'll wind up looking at an algorithm. Some of the ideas today I'm discussing came from American Academy of Physicians and Surgeons journal. Was an article by Herman Borg based on, or talking about evidence-based medicine, because evidence-based medicine really drives the algorithms that we're using for a lot of treatment standards these days. So again, in personalized medicine, we're looking a lot at screening and risk stratification and drug selection, but really it's been playing a very small role in treatment, on the treatment side. When we start looking at treatments and treatment algorithms, it's really the opposite of personalized medicine. It's actually in personalized medicine, because it's kind of a one-size-fits-all approach, and that people hopefully will fit into the algorithm, and the algorithms don't always give you enough latitude to make sound clinical judgments. The algorithms are based on epidemiology ranking. In other words, evidence-based medicine helps us rank data, or rank evidence, usually by committee, based on how the trials were done. So for example, if it was a good quality randomized trial, you get a very high grade epidemiology ranking. It's relatively free from bias. And of course, the lowest ranking is anecdotal. One unfortunate thing about that is that sometimes it kind of overlooks or negates the impact of anecdotal observations, which actually have been very important historically in the development of medical procedures and drugs. So, but in this system, the anecdotal and observational evidence gets downgraded very low. And that may be one of the weaknesses of evidence-based medicine, in fact. But it has helped develop these algorithms. And why were these treatment algorithms made? I mean, they're supposedly the pathways you're supposed to use to treat everyone, is kind of the implication. But their history is really a little bit of a different story from where they are now. Originally, a lot of treatment algorithms were described as paradigms for teaching and for helping with decision making on complex individual patients. But the emphasis is that it was used to approach individual patients. And now it's sort of been conveyed more into treating all patients, because a lot of algorithms have been expanded into clinical guidelines, and they're aimed at entire populations. I started to understand the impact of this through the maintenance of certification process. I'm a board certified urologist by training, and when I first started taking the board exams, every few years you have to review and take these tests, and I was being tested on what standards of care exist. But in the last few years, it's been very, very specific, and the standards of care refer specifically to the treatment guidelines that are accepted for those conditions. And they're very well defined. So that leads to another problem. Are they too defined? So one of the problems with algorithms is that sometimes when you define pathways for how to treat patients, that once they're written down and recorded and accepted as standards for the industry for which they train residents and do maintenance of certification, then you've replaced a lot of what was going on between the physician and the patient with expert protocols and best practices. These are not always the best practices, but then they become very compelling. For example, I called it the tail wagging the dog. I mean, these were supposed to be helpful to help us think through proper ways of evaluating complex patients, and instead they've become standards of care, and they can in fact sometimes force providers, which is the new name for physicians and others, and force providers to stay in compliance, which means obeyance with these rules. And the rules are generally made by payers, third party, or sometimes governmental agencies or peer review organizations. So that's it's almost like a dual-edged sword. These guidelines were meant to help us and help us think through these disease processes, and instead they can be used in a punitive way, because if you don't do exactly what the algorithm said, perhaps a malpractice attorney will say, well, this is, you've broken the algorithm, you've practiced below the standard of care. So in many ways, they are unpersonalized medicine, algorithmic pathways. Medicine's far too complex for simple algorithms, and they almost remind me of like the pre-AI computer programs of the past, whereas now we understand that the networks are much more complex than that. And it's easy to forget that most of these studies were done on people who didn't have multimodal problems and multiple diagnosis, and they were just in these large studies. And the fact that they had very specific genomes and proteomes has been greatly overlooked. And so there's this huge biologic individuality, and it's affected our evidence-based medicine studies because we've omitted some of the vast biologic individuality that we see in our patients. Again, one size doesn't fit all. So there are big issues with algorithms, and I call them unpersonalized medicine. Here's an algorithm that is how to treat interstitial cystitis. It's one of our more complex diseases that we deal with in urology. It's like chronic bladder pain. This is from the American Urologic Association. And I understand all the data and hard work that went into developing this, but it's really hard to use this in a real meaningful way in your clinic. And yet somehow, if you might break this pattern, maybe someone will say you practice below the standard. Another example from my world, urology world, of catastrophic failures of algorithms is there's been a big controversy over PSA screening for prostate cancer. And one of the most important age groups is age 55 through 59, and the current recommendation is that it comes down to that whether you should screen your patient or not comes down to shared decision making. If you poll, and this has been done, family doctors, hundreds of them, on what shared decision making means, almost no one will understand or agree. I actually did a little drilling on this and found this organization called Informed Medical Decision Foundation, and they actually list these steps of shared decision making. But the problem is it really doesn't work in real practice, because what does that really mean? I mean, you're supposed to tell your patient what's going on, be involved with him, inform him, give him choices, give him alternatives, and make a decision with him. Isn't that what we've always done as physicians anyway? So what that really is saying is nothing. So all the algorithmic thinking led to, oh yeah, just talk to your patient. Okay, so anyway, the main focus of why I'm talking about algorithmic thinking is it's playing such a large role in medical care now. And one of the visions that I have, and I think Mark shares, and a lot of the doctors we work with, is that personalized medicine will develop and get traction and become really the next major advance in medical care. We want to take it from screening and detection and risk stratification and chemo selection. We want to take it to the next level into the treatment realm. And the whole focus of personalized medicine is somehow matching a treatment to your genetics. So, if you're going to match a treatment to your genetics, what about taking that to the next level? And that's using your genetics as a medicine. So how do you use your own DNA to help yourself? And of course, the way we see that can be done, our vision is that that can be done through your own stem cells. Your stem cells are repair cells. Your stem cells from your body, not from some other source, but your stem cells have no other purpose on the planet but to repair you. And that's very powerful. So the idea is, how do you use them? And can you use them, the way we are looking at stem cells today is a lot like probably the way they looked at penicillin perhaps 100 years ago. They would give some, and then someone would get better, and they go, oh, here's a good treatment. But now we understand that penicillin is dosed, and then we understand the intervals, we understand the dose, and we've improved on penicillin. So that's where we're going with cell therapy. But at this point in time, if you want to bring cell therapy into the personalized medicine realm, you have to look at perhaps having a reserve of your own stem cells, because a one and done or a one-time treatment isn't always effective, and sometimes it just starts the process. So again, you want to use your own cells with your own DNA, no risk of allergic reaction or rejection, no risk of transmission of disease. You can use your own custom dosing schemes, which we're just starting to understand. Mark and I have treated over 6,000 patients with their own cells, and we're starting to realize that certain conditions do require certain types of dosing and certain repeat dosing schedules, and not everything's the same. Treating an autoimmune condition is very different from treating a knee, for example. And then the next stage would be industrialization, which means expanding this. And if you can create your own repair cells and have them available for what in military terms we might call a surge, this is really an advanced form of personalized medicine, because what it is having, what might take your bone marrow 6 or 9 months to create, you can have in a small sterile bag that you can give to yourself to deploy systemically or regionally, and you have a large number of healing cells, which theoretically would shift the healing equilibrium in a favorable way, just because there's so many of them. So this is what I call real health insurance. It's funny that health insurance, I often say health insurance is not really like insurance. It's a different kind of product. I guess catastrophic health insurance is more like insurance, but real health insurance is, maybe you could think of it in terms of having a large supply of your own cells that you could use in your whole lifetime for a lot of different things. Maybe we'll find out at some point that it mitigates aging, which is a form of chronic cellular degeneration. There's so many other degenerative conditions. The most common thing we treat, probably 80% of what we treated, is arthritis. That's probably the most common degenerative condition, but it doesn't have to stop at arthritis. Certain conditions, like we've seen chronic neurodegenerative diseases, really require surges or higher numbers of cells and repeat treatments. We're understanding that you can use cells to carry antibiotics or cancer-killing biologics into the cancers and into damaged tissue that's been damaged by cancer process. Your own stem cells may be useful for transmission of gene therapy. There's a lot of reasons beyond the scope of this talk, but there's a lot of reasons why your own cells can be incredibly effective in carrying, in many cases, viral vectors or other gene therapy systems. When you combine the repair system with the immune system, you can get a terrific combination. Repair system identifies damage versus undamaged. Immune system, of course, identifies self versus other, and when you combine them, you can get a synergy. And of course, the on the horizon that we're starting to see, but we're still not there yet, is growing things in the lab, growing organs and creating replacement parts. There's been a lot of attention paid to that because it sounds very exciting, but it may be that if you replace, if you are chronically and consistently working on cellular degeneration, you might not deal with as much full organ damage or that requiring full organ replacement. So sometimes it comes down to a numbers game. Basically, in your lifetime, you are on the planet exposed to a lot of bad things, and I listed a few: injuries, radiation, surgery, violence, infection, behavioral and lifestyle issues, overutilization, that could be sports or work-related injuries, genetic and epigenetic, mental and physical conditions, toxins, etc. Really, at the end of the day, it comes down to these things damage cells, and cell therapy might mitigate these. And that's just in terms of treatment. Then there's the entire other realm of prevention, which is another use of cells. You don't have to wait until you have one of the above things I just mentioned, but maybe you should do it as a mitigative or preventative cell therapy regimen, and that's something that I think people will be looking more at over time. And it comes down to, as I said, the numbers. It's a demand and supply issue. Any economist would understand this, that human repair cells generally go down in quality and time, quality and number over time, but really our demand, because of being exposed to life, our demand goes up, and there's a mismatch there that we might be able to mitigate by having a huge laboratory-based supply of our own cells. Mark and I have started to develop the Cells on Ice program, which is looking at safety of freezing your own cells, and looking at ways that we could develop them for use in large numbers. It's basically a simple liposuction-based procedure because it's adipose-derived cells. The cells are converted to SVF. in a sterile environment and then frozen, and then these cells can be used to be expanded or plated, and you can get hundreds of millions of them. We've characterized them, and they're all basically CD73, 90, 105 MSCs, and you can put them into very, very high doses if you feel that that would be indicated for the patient, and they can be delivered right away to patients on an as-needed basis, and we can use them as a bioinsurance. So we started looking at this under IRB. We've done some transport and safety studies, which Mark, who's right behind me, is going to start talking about, of what those studies look like and how once all those studies are completed, that we will be able to help people in the future have or achieve true personalized medicine on the treatment side. So I'm not going to take questions just now. I'm going to turn this over to Dr. Berman, and then we'll take some questions at the end. Thank you very much.
Great. Hey, thanks, Elliot. That was great. Now you see where we're headed. We're in a new era of medicine. Whether you like it or not, you've got cell as replacement parts. And I think it's clear what Elliot was talking about is this is a numbers game. We eventually run out of cells. We've been talking about this for a long time. That's why we coined the term CRT, cell renewal therapy, with the idea that if you have enough replacement parts, you can keep that heart ticking for a lot longer than you expect. And we're hoping that's what we eventually can get to. Right now, what are we treating? We're seeing all these people with chronic conditions. In fact, I think we're treating the most difficult problems with cell therapy right now. So if you think about it, you get people who've tried multiple treatments, and especially orthopedics. They've been on NSAIDs and steroid injections, hyaluronics, they maybe even have surgery, and then finally they say, well, before you get a joint replacement, let me try that crazy thing you're doing with the cells. So luckily with point of care, we get enough cells, especially with that, we get a tremendous number of cells with SVF that we can affect some good positive results with point of care treatment. So that's pretty exciting. We can get somebody within a month maybe having knee arthritis go away and pain go away. Hey, listen, I'm a personal patient now. Dr. Lander did liposuction on me, a mini liposuction. He treated my sciatica. Well, actually, Dr. Feller treated my sciatica with a little CT-guided injection, and it cleared up in about a month. It was terrific. I actually had an opportunity to expand some of my cells, put them on ice and freeze them, and then I developed another sciatica on the other side from a soul cycle class my wife got me to go to, and it took about a month with the expanded cells, and we were able to clear that up. So we're going from the exciting part of the talk that Dr. Lander just gave to this part, which is more of the nuts and bolts of how the Cells on Ice program works, so you understand what we're trying to accomplish. And this is really pretty simple stuff, but you have to understand, the FDA is mandated, and pretty much it goes back to the 1944 Public Health Service Act to prevent the transmission of communicable disease. So everything we do is working under an FDA-registered cell bank so they can properly take your cells, make sure they're pre-screened for any serious infectious disease, and then screen the fat that they can process into SVF and eventually plate it out and grow into expanded cells. And all this is validated from start to finish for sterility, so that when we get it back as doctors, we can evaluate it. In fact, we already did a transport study in over 20 samples, so every sample was shown to be completely sterile. We sent the cells to the lab, and we showed we had good flow cytometry. In fact, we could evaluate the cells that we got, and we could show they had 70, 93, 105, all the proper markers for stem cells, or 73, 90, and 105, excuse me, and all the proper markers. And we know what we got is what they said we got, we actually did get. So here's how it works on a step-by-step basis. Basically, you're going to be able to go online pretty soon. By the end of February, you do all this stuff. Right now, you have to start out with email and fax. But we take the doctor, we create a username and password, and basically what the doctor has to do is evaluate, read these things. Read the cover letter that you're going to get. It's a simple cover letter that explains what we're doing. You have a complete standard operating procedure, so you know step-by-step everything that's going on. And then you have to fill out a practitioner agreement that you can sign and send in, and this only has to be done one time, and that goes along with your physician registry. So once you're registered, you're all set to go. Then you have to get the patient involved. Now, one of the most important things on this list is the infectious disease clearance. The patient needs to, what's that? Okay. Doctors are registered as sub-investigators. So basically, everybody who gets registered, you're part of a sub-investigator. I think what Dr. Lander was handing me a note was to point out, everything we're doing is under IRB approved protocol. The basic IRB protocol we're working on right now is for the first 100 patients for patient safety. And we had to go through hoops to get this IRB approved, but we have it appropriately approved because we've done all the proper validations. So anyway, excuse me?
For CSN doctors only.
Yeah, this is all for doctors within the Cell Surgical Network organization. And so everybody who's in CSN has already been properly validated and also is qualified because they're doing the procedure all the same way. Once this can expand, we'll hopefully teach other doctors how to do this, but we have to go through each step and do it the proper way. The next step, though, is our patients need to have some paperwork. There's a lot of paperwork involved with this. They'll need informed consent, a client agreement, and authorization to release their cells. So the infectious disease clearance is pretty important. We're looking, the FDA mandates that you have to screen all your patients rather, for HIV, hepatitis, syphilis, all these particular diseases, even cytomegalovirus. And we'll go through that. So your patient's going to get informed consent. We have them take this information home and read it. There's a lot of information. They should have plenty of time to read it before they come to the office and sign this stuff. There's a client agreement. So they're going to be sending their fat to a laboratory. They have to understand what's going to be done with those cells and what's involved, and they need that agreement signed. Finally, they need to get their cells back. Any patient who's alive and healthy can call for their cells and have it sent to their physician anytime they want to. But what if something happens to the patient and they need somebody else to get their cells? We need to have proper authorization from people approved by that patient. So your wife, your husband, your kid, whoever it is may be, authorized to get your cells if it's an emergency. Your patients will get a form similar to this. This is only partial form, so you get the idea. The infectious disease clearance, and they'll take this to a lab. They'll have a blood test drawn. It takes a few days to get these clearances. So this is usually done within a 2-week period, 13 days before collecting the cells. Once you know the patient is properly cleared for infectious disease, you can go on to the next process. And that's having the patient understand, what's the process. We have a basic plan where they can send fat to the bank and they'll separate the cells out of stromal vascular fraction, freeze it. But for a little extra money, they'll be able to store as bioinsurance. And you could go all the way up to getting 100 million cells every month, potentially, for a certain amount of money. And so we have these plans. This is all online. There's nothing you have to memorize. It's just that you have an idea of what's going on. Obviously, there's a payment involved, and we get that information as well. Now, you have, once you get all this stuff together, you're going to get a cryo kit from the bank. And basically, this allows you to put the cells away. So here's what you're going to get. Basically, there's a transport medium, comes in a sterile bag. Basically, you're going to put the patient's name, information on it, a little bit of information so it's identified. There's a port. It's very simple. This middle port that looks a little unusual, but it has a valve on it. And what we have to do is wipe off the valve, make sure the valve is completely sterilized, and then you can open up the port with this release. The cells that you've collected, and we compress, so we get maximum compression, we get rid of the local anesthesia, can then be introduced directly to this port. And this is done in a sterile manner, so you got to make sure you're not touching that port or doing anything to contaminate it. And then you're basically just introducing your cells right through the Luer lock into the bag. This bag is then going to be properly packaged in a biohazard bag, and there's a insulated bag that it goes into. You'll notice there's a thermometer in it that's on a battery operated, so it logs all the temperatures, so we know that we're properly maintaining a proper temperature. This can go by room temperature, by the way. And we've put this thing on airplanes. We've done all kinds of validation studies. We know the cells transport really well. And once you fill out the rest of the paperwork, goes in this box, and you send it back FedEx to the bank. Now, once the bank has the opportunity to take the cells, and say you've requested 50 million cells for deployment, then the cells are being expanded in the bank. This can take 4 to 8 weeks, and then you have more paperwork to do so you can get the cells back. There's a lot of paperwork involved with this, but we have to follow all these steps so we can assure the patient they're getting their cells back and the cells have been properly handled. As I said, the bank will take samples of cell and they'll validate sterility from the start right to the end. So when you're done, you're going to know you're getting back sterile cells. So there's an IRB protocol for handling the cells that's available. We have that covered, and everybody will get copies of that who is participating within our network. There's an investigator's agreement. So once you agree that you're going to be giving back these cells to the patient, you also need to put this agreement, sign, and return it to us. All those agreements have to be done just one time. You don't have to do it every time you do a case. The informed consent is for the patient, that they should get this well in advance so they know they're getting back expanded cells and they know what to expect, what the risks are, and they have the option to opt out or get their cells back. Also, because we're doing this as an investigation under an IRB, we want everybody to have a CBC and Chem 14 or Chem 20 panel done in advance of the procedure. If you have the labs done up to 3 months prior to the procedures, those will be okay as a baseline study. 3 months after the procedure's been done, we're requesting everybody gets a CBC and a Chem panel again, so we can chart if there's any variations in their labs. Now, on top of that, the CSCTC, the primary investigators, myself and Dr. Lander, we're also sending the cells back to our labs for flow cytometry for further evaluation. On top of that, we're recommending that you also send out a culture to your lab, and hopefully in the future we'll be able to culture these things right away. So when you request the expanded cells, there's a work order that you have to fill out. You also have to show, what's the basic plan or the particular plan your patient wants. And this is all really pretty simple. And by the way, we have people in the office who will help you with all this stuff. Once you get a request to retrieve the cells can be sent back to you. They'll come in a little container like this. This is a sterile tube. And once you open it in a sterile manner, use something like an 18-gauge spinal needle to remove the cells. Again, we recommend taking a drop, sending it out for culture. It's a good idea to count the cells and validate the number. We've been doing this over and over again. What the bank tells us they're sending us, we've been able to validate that's what we get. And what's really exciting, like I said, when we send the cells to our labs at UC San Diego, they've been showing us that, of course, they've got CD 90, 73, what are my numbers? 105. It's really interesting. With SVF, the typical markers are CD 34 and 45s. When you grow those cells out in the bank, they convert to MSCs. So you have pure MSCs with all the markers, the 73, 90, and 105s. So anyway, this is really pretty exciting for us. Like I said, we will also do some other validations, and we hope to be able to, Dr. Lander particularly has been looking at devices so you can do instant checking for infectious disease, particularly gram-negative, gram-positive anaerobics, things like that, right at the time of surgery. And this will be helpful for our SVF cases going forward as well. And we think we're right there, but we'll let you know as soon as it's available. Finally, you'll have a deployment report. So this is an op note that you'll fill out. And again, this will go online. Everything we're doing is being registered online. Fundamentally, we think this is what the FDA should be doing. The FDA really has the duty to make sure we're not risking any disease transmission. We know we're not. We're cleared for infectious disease. We got complete validations of sterility. The lab's registered with the FDA, and fundamentally, we've been trying to work with the FDA and set up our own IDE studies for knee arthritis. We have a pretty good relationship with them. We're not making claims. We're not trying to sell a drug. We're using your own body parts. And we're following really the scope of the law in every regard. Having said that, the real important thing for us is to get the data. So we have IRB-approved protocols. The first 100 patients will be evaluated on safety data. You'll put this information onto an op report, and then this will be filed on our database. Once we have all this data, we can then look at it. If we have any serious problems, all the adverse events are going to have to be reported to the IRB promptly within 24 hours. If there are no serious adverse events, we'll look at all the data and we'll correlate it and we'll get back to you. I'm sure it's going to be very exciting. So anyway, we're glad you joined us for this webinar. We're able to take some questions, and if Elliot joins me, we'll go over those questions right now. By the way, one last comment. We have a meeting, the 4th Annual Cell Surgical Conference is going to be at the Beverly Hilton Hotel, June 2nd to 4th. The 3rd and the 4th are going to be didactic days, and we look forward to seeing as many of you as want to attend. We think this year is going to be, every year has been exciting. This year should be as exciting as the previous years. So thank you for joining us on the webinar. What questions do we have? All right. Here, come on up.
Thanks for joining us. Some of these questions were submitted in advance. Some are just coming in. Maybe we'll take a couple of these. Okay. Are adipose cells more effective on joint tissues, or could they be also effective in delivering cancer cures? Stem cells are useful for certain cancer treatments. I don't know about the word cure, but stem cells, as I mentioned earlier, can be very effective in carrying certain biologics to cancer. Stem cells exhibit a property called tropism, which means they're attracted to cancer, probably in essence being attracted to the damage that cancer causes. Most of the work we're doing at this point is regenerative, and we use adipose cells for a number of reasons. The adipose stem cells are very robust. They seem to be very healthy in terms of their ability to repair things and divide and multiply, even into your late age, and even if you have systemic disease. The cells in your bone marrow seem to be more susceptible to age and chronic disease. And also, the stem cells from fat are very easy to obtain. They're just under the skin, and it's relatively painless to just remove a few versus drilling into bone marrow. So all the work we do is based on the stem cells from fat, we have quite a bit of experience now and a lot of safety data. We also use them intravenously, and there's no safety data for using the bone marrow-derived cells intravenously, but I believe they're probably safe intravenously also. But we have thousands of patients now with, that we treated IV with good results. We have no problems. We have a safety study coming out in the American Journal of Cosmetic Surgery in March on our experience with our first couple thousand patients, showing remarkable safety.
Yeah, it's a good point I should have mentioned. Yeah, this is a peer-reviewed journal. We submitted to the American Journal of Cosmetic Surgery because I was the past president of that academy, and I thought it was a nice thing to do. Having said that, they didn't give us an easy time in submission because they didn't see the cosmetic relevance. We had to point out that you could use these cells with fat for breast augmentation, and also because we're doing liposuction, that's how it started. And a lot of this stuff started with plastic surgeon and cosmetic surgeons, so that's why we submitted to that journal. Let's see, what else do you want to talk about?
You want to answer this one? Do the expanded cells include any SVF?
No, the expanded cells, if you heard what I said, SVF, you'll see cell markers in SVF that are mostly 45 and 34. When you expand them, all the cell markers are typical mesenchymal cells, which are going to be 73, 90, and 105. So yes, you're going to get a pure line of cells when they're expanded.
Okay.
I can't even read this.
Can stem cell therapy benefit stroke patients after almost 2 years? Mark, do you want to address that? Because I think that's a case where multiple treatments could be useful.
Well, you know what? That's a good question. We have a stroke patient right now who's 92 years old. He had a stroke 10 years ago. And we've given him a lot of treatments with just SVF. We're hoping we can get expanded cells back so I wouldn't have to keep doing liposuction. But the answer is his family is convinced, and I can tell, that he's getting better in spite of the fact that he's had a stroke 10 years ago. These are very difficult things to treat. It's probably worth noting that while we're treating these very difficult chronic conditions, cells are probably better in acute conditions. And we've got some real interesting information to kind of prove that. In fact, my son Sean, who's actually right here with us, did a great study on concussion, and he showed that if you gave rats cells right at the same time as concussion, you completely mitigated the problem. It makes sense because you've got the most cytokines and you've got opening of the blood-brain barrier, you've got a good opportunity to treat the problem. So the same thing with stroke. We had one of our patients, and this is obviously anecdotal, but had a small stroke with a couple small bleeds, and we gave him cells within a week of the injury, and within 2 to 3 days he was feeling much better. His wife could tell it was like night and day. And so, that's anecdotal. There's no double-blind study to prove that what we did was successful for him. But I think we've had enough experience, and anybody who's been doing this for a while has a pretty good idea that the closer you get to acute injury, the better off you are with your cells. Fundamentally, this is like the oldest form of medical treatment. And I really think we're in a whole new era of medicine because every injury we have is a cellular injury. And everything we've been doing up to now is to, try to treat the condition that's causing the injury and allow our bodies to repair itself. And now we really have a lot of repair parts. And stem cells have got to be the best repair part you can have for any particular injury in your body. So we know that. We need the data, and we probably need some more studies to, prove it to the people who need the proof. Our patients don't need a lot of proof because they're getting better. What else do you have in here?
How many maximum passages? We're going to P1 mostly.
Most of our passages right now have been between 2 and 3 passages. What we are also asking everybody to do is when you get the cells back, you'll have a number of doubling times as well as a number of passages. So we'll be able to see the correlation between doubling times and passages if there's any fundamental difference. In most studies, you have to go up to 13, 14 passages before you start getting any abnormalities in the chromosomes of the cells. And that's been pretty well documented. In fact, there have been studies going up to 30 passages where even though there's some abnormalities in cells, there haven't been any evidence of cancer causing from the cells or anything unusual. The fundamental thing is what Dr. Lander was talking about is this is personalized medicine. And this is where we have an opportunity to really deal with, the problem. Now, having said that, we can eventually get to great dosing. The problem, I think, is going to be there's always going to be a variable in how many cells have been injured. With orthopedic stuff, we have guys like Centeno that's done some great work and shown that you need maybe 3 to 5 million cells for your average knee arthritis. But, for what do you do for neurodegenerative disease? We don't know how many millions or billions of cells have been injured when somebody starts getting symptoms, and especially if somebody goes along and is progressive. Interestingly, in that department, we had a patient, Dr. Lander and I, who had optic neuritis, and she was on television, so she was very popular and very well known about. But we literally gave her 32 almost monthly treatments of SVF
for free.
For free, yeah, thanks. We're a real money-making business. But we gave her a lot of treatments, and basically she would get better and then fall off a little bit, get better and fall off. But it's been over a year now, and she's been stable. This is a woman who came into our office blind on 40 milligrams of prednisone, who now has a driver's license and can see and hasn't needed a treatment in over a year. So it may be that we finally got enough cells into her that it stabilized her problem and she did fine. Hopefully, we'll see. And this is what we're learning. You may need, expanded cells may give you the opportunity to get to these conditions a lot better. We've seen that with some of our colleagues who have used expanded cells. And we know that this is probably what's going to be necessary, especially for these difficult neurodegenerative conditions.
There's a question about cancer growth and cell viability when they're frozen. In terms of viability, most of the cells survive the process. We spent almost a year doing a transport study, which is just receiving multiple doses of expanded cells and making sure that they were viable in different transport media, making sure that they were completely viable and that they were sterile. And we did that on dozens of patients. And in terms of cancer, we continue to follow that. We addressed that in our safety paper. There's been no evidence of any cancer growth associated with adult mesenchymal stem cells, and we continue to follow that. In our first couple thousand patients, the incidence of cancer was lower than in the general population. So we feel that our findings are in concurrence with the work being done by every other group around the world that's found not a shred of evidence that adult mesenchymal cells is related to cancer. I just want to emphasize something Mark said about the doctors in our network. This is not something that's any doctors can order or any patients can order. This is only under our IRB protocols for doctors in our network who've signed up as sub-investigators for this particular study. And at some point after we collect some basic safety data, we will probably submit this for a regulatory approval so that it's something that we could market to patients all over. But I think we need to get our basic safety studies done first.
Yeah, I think it's really important. I want to go to the FDA if we're going to do a study, and at least say, hey, we have enough evidence this is safe. I'm not sure what we're ever going to make claims about. Fundamentally, your cells can fix everything in your body that's broken by a cellular problem. And we'd end up making claims about almost every problem and disease in the world if we were going to have to sell your cells to yourself for particular problems. We got a question here about efficacy of SVF versus expanded cells versus umbilical stem cells. The answer is we don't really know that one cell's better than another, but we've seen very good results with SVF, and then we've seen the same good results with expanded cells. It almost seems like these are, picture an envelope with all your DNA in it, and regardless of whether it was SVF or expanded cells, it's kind of like a blank slate of your DNA. And to me, I've always felt, and I can tell this based on certain clinical issues, that the message coming from the injury is really more important than the cell. The cell's blank slate your DNA. The message, the signal from your injury dictates what it's going to become. And an umbilical cell should work as well as any cell. And so fundamentally, umbilical cells are adult stem cells that have been gathered and cleaned up from donors.
But they're not your own.
They're not your own.
That's the main thing.
So there are going to be people who need allogenic cells because maybe they don't have enough fat, or maybe their bone marrow has been injured from disease or who knows what. And so there's going to be a place for allogenic cells. I think we need to continue to keep our minds open, investigate these things, and as we get more data and we're better able to introduce this stuff, then that's where we'll go. I'm sure sooner or later, there's a whole bunch of companies trying to make cells in a bottle, and we think that's going to be great. Of course, we really believe that ultimately, if you have your own line of cells, you'll have the cells that won't be rejected. It's your own DNA. You're not going to have to worry about building up immunities over a period of time. We really believe that CRT, cell renewal therapy, is going to be the answer to anti-aging medicine. Anti-aging medicine is everything you do to keep your cells living longer. CRT would be the replacement parts. So fundamentally, there's no reason we shouldn't be able to stay alive well into our hundreds, 150 and beyond, as long as you have replacement parts. I mean, ultimately, one day, why do you not wake up? If you don't get run over by a bus, you run out of enough cells to conduct a heartbeat, and you just don't wake up one morning. But this could give you enough cells to keep going indefinitely. Let's see, what else do you have? Okay. Anyway, I think it's time that we call it a day. This has been our first webinar with you. We really appreciate you coming on board to join us. We'll be happy to answer questions that you might give to us later on, and we'll write back to you. And the most important thing is thank you for attending. Let us know what we can do to answer your questions, and we'll try to get to that in writing. And maybe one time we'll have a webinar, we'll just take before and after questions, and we'll see what we can do with that.
Thank you.
Thank you.